Hello everyone and welcome to Instant Biology by Dr. Neela. Again, today I am here with you with another very important and interesting topic, and this topic is vaccination or vaccines. Majorly this topic we have already studied in Hindi in the previous lecture, but in today's lecture I would be aiming to introduce this topic, to teach this topic in English, because many students have requested me to start teaching in English as well, so that these lectures can be of use to them as well. So without wasting much time, let us start with vaccines or the concept of vaccination. Now you can see that this lady is or has got vaccinated and she is very happy about this. Now what are vaccines and what actually is vaccination? Let us start with this. Now vaccines are, or vaccination is, a biological preparation. Basically vaccines, what are those? These are biological preparations. Now what do you mean by biological preparations? These preparations or these vaccines can be anything that are related to the virus, okay? Because they or these vaccines are aiming at giving us proper protection and activating our immune system and not actually causing harm to our body, but just activating it so that when the actual virus or when the actual pathogen comes to your body can be very well prepared for that, and it can cause or it can mount immune response against that particular pathogen. So this is the concept. So it is a biological preparation. You would get to know more about this term biological preparation in just a bit. It provides active acquired immunity to an infectious malignant disease. Now what is the purpose? As I've already said, that the purpose of this particular biological preparation or vaccine is to provide an active acquired immunity to an infectious malignant disease. Now infectious malignant disease means that can cause an infection, that can cause problems inside your system. And this particular vaccine, what will it do? It will provide active acquired immunity. What do you mean by active? By active, I mean that when you are injecting the vaccine inside an individual's body, that body itself will start producing immune cells against that particular vaccine. Therefore, I am saying this as active. Now, active simply means this only, that because of this vaccination, the individual's body will start to produce the immune cells, will start to produce, let me just write down, they will start to produce the immune cells. The body will itself start to produce the immune cells. That is why we are saying that as active, and this is acquired. What do you mean by acquired? Because you or the individual has acquired it in its lifetime, has acquired these kind of cells in its lifetime. These cells were not present since the very beginning or since the birth, but these kind of immune cells have been generated inside the body after the infection or after the insertion or after the administration of the vaccine inside the individual's body. Okay, so this is the concept of active and acquired. You should understand each and every word very carefully. To an infectious malignant disease, this I have already told you. Now let us understand that what is the history or where does this concept of vaccination actually arise from. So the first individual or the first gentleman who basically gave this or administered this kind of vaccine was Edward Jenner. What did Edward Jenner do? Edward Jenner actually inserted or administered a vaccine against, basically he was testing protective effect of cowpox against smallpox. Now what is the storyline behind this is that during the time when Edward Jenner was working on this immune cells, basically there was a huge problem of smallpox that was arising at that particular time. Now people were dying because of this smallpox, and in the same time period there was another problem, there was another disease that was happening with the cattle, with the cows. It was seen that the cows were getting infected with the cowpox disease. Okay, and Jenner also saw that the females or the ladies who were milking the cows, they also developed very small rashes on their body that were type of cowpox disease only. But since cowpox happens only in the cattle, the effect was very minimized in case of the human beings or in case of those ladies. Okay, but similar to cowpox, there was another disease that was very prevalent at that time, and I told you about that. That disease was smallpox. Smallpox was very pathogenic, very virulent, and it caused a lot of deaths during that period. Now, Jenner observed that if he prepared what do you say, biological, if he made a biological preparation, and in that biological preparation, what did he do? He took the help of the cowpox virus, or he made use of the cowpox virus and inserted it inside the body of patients. Then what happened is that because of the similarity of the cowpox and the smallpox virus in structure, the immune system of those individuals, they started developing immune cells against that cowpox virus. Now, as I've said that the cowpox virus is very similar to the smallpox virus. Now, when eventually the smallpox actually infected that individual's body was very much ready with the immune cells, and it fought very well with the smallpox virus, and that individual did not die. So this was the concept that Edward Jenner made use of, and that is why Edward Jenner has a very important role that he plays in the story of vaccination. So I have already told you about Edward Jenner. It is related to the protective effect of cowpox against smallpox. You should be very well aware about this. Okay. Now, what are the properties that a good vaccine should have? Let us understand this. What are the properties that a good vaccine should have? A vaccine should be able to induce B cell response. You already know that what are B cells, okay? You already know what are T cells. So a good vaccine should be able to induce the response of the B cells as well as the T cells, these and this. Apart from that, the vaccine should be able to the response that a vaccine generates should be very long-term response. That means it should not be very short-term response. Once you take a vaccine, it should be for lifetime, or for, let us say, 15-20 years, so that you do not get to be jabbed again and again and again and again. Next, side effects. The side effects should be as low as possible. Now, you might be getting an idea, you might be having an idea about the COVID vaccines, right? Many individuals have complaints regarding the COVID vaccine that they have developed some kind of a skin infection after getting jabbed from a Covishield or Covaxin. Now, with that point only, I am trying to say that the side effects of a particular vaccine should be as minimum or as low as possible. Next, memory cells generation. Now, a good vaccine should always generate high number of memory cells. What are these memory cells? These memory cells will keep the information that is related to how this particular infection was eliminated from the body. All the information related to that is kept inside the memory cells, and the basic significance of the memory cells is that only when the infection is again, when again the infection comes, then these memory cells can be used they can be used again for fighting against that particular disease, and this would be done in a much shorter time as compared to the primary infection. When the first time the infection would have come, it would have taken a longer time to eliminate it, but because of the memory cells have generated after the first infection, when the infection comes the second time, because of the memory cells, because of the information that is present inside the memory cells, This secondary infection would be eliminated in a very short time as compared to the elimination of primary infection. So this is the concept of memory cells. Now let us move on to the next slide. Just a minute. Okay. Now let us come to the next slide. Over here you can see that most effective vaccines till now are polio vaccine and smallpox vaccine. Now these are one of the most effective vaccines that have been generated up till now. But what we still require is that there are multiple other diseases that we do not have vaccines for. For example, malaria, tuberculosis, AIDS. These kind of vaccines have still to be developed. We do not have vaccines for these kind of diseases. Also, there is a requirement for improvement of the already existing vaccines in terms of safety efficacy, in terms of cost lowering. Now, a vaccine should be in reach of the common people, right? Therefore, all the efforts should be put in to bring down the cost of the vaccine to the reach of the common people. Now, let us move on to the next topic, next slide. Just a minute. Yes. Now coming to the types of vaccines. We would be talking about all the different types of vaccines, but in today's class I would be aiming at talking about the live attenuated vaccines. Now what are live attenuated vaccines? In case of live attenuated vaccines, you use the live organism or you use the living organism. So in this case you are making the use of living organisms. Okay. Now when you are using the living organism, obviously your body or your immune system would generate a very robust response. What do you mean by robust? A very high intensity response would be generated by your immune system. But there is a massive risk involved. Now what do you mean by when I say risk involved? Now you understand that we are using a live organism. Now this live organism can always revert. What do you mean by reversion? You are using an attenuated virus, attenuated organism. Attenuated organism means weakened organism. That means it has the capacity to grow, but it does not have the capacity to cause disease. That means it can grow, growth is possible, but disease is not possible. Growth is possible, but disease is not possible. This is what we mean by attenuated. Okay, so risk is involved because when the body this infection or when the virus or when the organism gets inside your body, it can cause reversion. It can again become virulent. It can again start to cause diseases. This is the case that can happen. So this is there is a risk involved that the pathogen can again get reverted and again start to cause disease inside the individual's body. So this is what we mean by risk involved. Now let us talk about attenuated microorganisms. This I have already told you. What does attenuation means? Attenuation means disabled or weakened. By disabled or weakened mean that the organism can grow, the organism can multiply, the organism can reproduce, but the only thing that the organism cannot do is to cause disease. Okay, so what is the concept over here? The microbe is slow transient. Microbe can have or can show slow transient growth, but pathogenicity is lost. That means the microbe is no longer pathogenic. It cannot cause any disease. Okay, so this is the concept. I believe this you would have understood. Let us move on to the next slide. Okay, just a minute. Natural attenuation, basically I have already told you about this, that the natural attenuation primarily or the first time was introduced by Jenner. He told about the natural attenuation process. This was a vaccinia virus that he was talking about. He inoculated vaccinia virus inside the humans. Now, what do you mean by vaccinia virus? This is the same virus as the cowpox virus, basically. So, vaccinia virus was the cowpox virus. Because cowpox virus can cause diseases in the cows only when it is inoculated inside human body, it will not cause disease. So, that is what I have written over here. It does not cause cowpox, but it causes immunity against the smallpox. Now, why is this? Because of the similarity of the structure, right? Similarity. Because of the similarity of the structure, this happens. This can be called as cross reactivity also. Okay. Now, let us talk about the next thing, attenuation. Let us talk about the process of attenuation. Attenuation, pathogenic bacteria or virus grown under abnormal culture conditions. Now, I have already told you what attenuation is. Attenuation, we have already said that it is weakening. Weakened. Okay. Attenuated means weakened. So, the microbe is already weakened, but how do you weaken a microbe? What is the process behind this? The simple process is that you start growing the microbe in a abnormal culture condition. What do you mean by abnormal culture condition? Now, whatever is required by the microbe for its normal growth, you are not providing it. You are providing it bad culture conditions. You are not providing it optimum culture conditions. Because of that, definitely the pathogen will have to change something or the other in its body, or obviously something will change in its body. Now, suppose you, let us take an analogy for this. Suppose you have a habit of living in the plain area, right? That means suppose you live in Delhi, you live in Kanpur, or you live in Lucknow, that is the part of the northern plains. But if you are taken from the northern plains to a place, let us say in Ladakh or Leh, you are not very used to living in that particular area. What will happen? Okay, you will have to make changes inside your body, definitely, or you will be bound to make changes inside the body. Therefore, the same things, the same kind of things will be happening with the pathogen or with the microbe. The microbe will have certain changes, will have certain things changed inside its body. The genome will have certain things changed. The same thing will be happening over here also. Once you grow a microbe in abnormal conditions, what will happen? Now, what are the abnormal conditions? Let us first of all talk about this thing. What are the abnormal conditions? Let us first of all talk about this. The abnormal conditions can be you are growing the microbe in a changed pH, you are causing radiations to the microbe, you are heating or freezing, changing the temperature basically, you are using the metal ions, or you are agitating it, you are causing the dehydration, some things like this. These are the different ways in which you can cause the abnormal conditions of the virus or the pathogen basically. So because of this, what will happen? Once this happens, the pathogen or the bacteria will have to change its growth pattern, or will cause changes, or it might not cause the diseases, it will lose its pathogenicity. And this kind of the pathogen or microbe can be given through intranasal way, can be given through intramuscular way, can be given through intradermal way, or can be given through oral ways. So these are the different administrative routes through which the pathogen or the microbe can be administered inside the body of the host. Okay, so this is the concept. Let us move on to the next slide. I have a particular case study in which I would be telling you how exactly, what is the way in which exactly the attenuation is caused. Now, I have the example of Mycobacterium bovis. Now, you might be knowing about Mycobacterium bovis. This, genera, Mycobacteria, is a very potent, or it can cause multiple diseases. You might be having an idea about Mycobacterium leprae or Mycobacterium tuberculosis. Both of these cause leprosy and tuberculosis respectively. These are very, very potent diseases. Mycobacterium bovis belongs to the same genus. Now, this For example, is Mycobacterium bovis. It is virulent. It can cause disease. It is pathogenic in nature. Mycobacterium bovis has other name which is also called as Bacillus calmette guenin, or it is also called as BCG. Now, when you allow this pathogen to grow for 13 years, mind it, we are talking about 13 years in increased concentration of bile. Now you would understand that bile is not a very good thing to grow upon, definitely, and you are growing this microbe on the bile for 13 continuous years. What will this lead to? It will lead to generation of the non-pathogenic bacteria. From the pathogenic, you are transitioning towards the non-pathogenic bacteria. Because of this, what will happen? The disease causing ability will not be present inside the the bacteria, right? Disease causing ability is no longer present in this bacteria, but it has the capacity to grow. It has the capacity to grow. Disease causing ability is no longer present, but it has the capacity to grow. So this is the concept, or this is the case study regarding Mycobacterium bovis. It is also called as BCG. So you should be knowing about this. Let us move on to the next slide. Okay, so what are live attenuated vaccines? This is another just a brief write-up in which you can understand that what is live attenuated vaccines in a short way. You can see over here that the harmful viral genome is present over here. You can see that this red in color, this is harmful viral genome. What you are doing is this harmful viral genome is broken down, or it is you have made changes to the harmful viral genome. So this is harmful, this piece is harmful, but what you have done is you have made mutations in the genome so that the disease causing ability has gone considerably down. Now this becomes weakened vaccine genome. Now you have weakened the virus by making changes inside the genome. So this is again a way in which you can cause attenuation. You have made changes in the DNA so that the virus can get attenuated. This is another way. Now again this particular thing, or this particular virus, can be inserted inside the body of the host to activate the immune system of the host. So this is the concept. Sorry. Now whatever I have just said in the previous slide, I have already said that attenuation can be made through multiple ways, and genetic engineering is one such way, right? We have just seen that you can remove the genes, the virulent genes, and therefore you can end up in a bacteria, in a virus that can be non-virulent, that can grow inside an individual's body, but it would be not causing the disease. Now multiple viruses or multiple vaccines have been made out of this concept. For example, herpes vaccine for pigs. In this, thymidine kinase gene has been removed. Now this is again a virulent gene. This was again a virulent gene, and this gene was removed, and therefore with this removal of the gene, the herpes vaccine was produced, and it was very potent in case of pigs. Again, another example is influenza virus. The influenza virus can be attenuated by means of temperature changes. If you increase or decrease the temperature, again the influenza virus would be attenuated. Let us move on to the next slide. Now this is the most important slide that you should be, just a minute, that you should be aware of, because in GATE examination or CSIR examination or DBT examination, most of these questions are asked. Now you should be knowing that what are the examples of the live attenuated vaccines. For example, varicella zoster, mumps, rubella, measles, BCG, smallpox, yellow fever, oral salmonella, and oral polio vaccine. Now these are the vaccines that have been made out of this particular concept. You should be remembering the names because this is very, very important. Okay, so now let us move on to the next slide. What are the advantages of this vaccine? Let us talk about this. Let us quickly go through this. What are the advantages? Okay. Now, advantages is prolonged immune system exposure to antigen. Now, what does this means? Prolonged immune system exposure to antigen. By this, I mean that this kind of a vaccine is causing delayed release of antigen. That means inside the host body, the antigen would be released very slowly. Now, if it is released very slowly, the antigen would be present for a longer period of time inside the body of the individual. The antigen would be present for a longer period of time. Now, when it is present for a longer period of time, the immune system would be activated again for a very longer period of time. Immune system would also be activated. Now, this is what I mean when I say prolonged immune system exposure to antigen. Okay. Now, immunogenicity would definitely be produced. The immune cells will be activated. Immune system will be activated. Highly effective memory cells. Now, this is happening, or the highly active memory cells are being produced because of the first point only. When the antigen is present inside the body for a very long period of time, higher number of memory cells will obviously get produced, and these memory cells will be helping in eliminating the pathogen if any kind of secondary infection arises. Secondary infection arises. This is the concept behind this. I hope you would have understood this particular concept. Let's move on to the next slide. Yeah. So this basically, as I've already told you, that this is such a potent type of a vaccine that you just require a single immunization. That means you would just require a single shot. You would not require multiple shots. Just a single shot is required. Only single immunization is required. Now, what will happen? The pathogen, as I've already said, that it has the capacity to grow inside your body, but obviously it does not have the capacity to cause disease. So replication of the organism or the growth of the organism will take place inside the body of the host. That is what is written over here. Inside the host cells, the replication will take place, and now you would have a clear idea about what happens inside the host cell. Now if a host cell is present, right? If the pathogen is entering inside the host cell and the pathogen is multiplying, the pathogen is growing inside the host cell, now it will obviously generate T cell response, because T cells are particularly targeted against intracellular pathogens. Now, if the pathogen is growing inside your cell, then obviously T cells will be activated, T cell response will be created. Now, I am again talking about a case study. This is very important with respect to this kind of a vaccine. This is oral polio vaccine. Now, you might have seen the different advertisements about oral polio vaccines, and you would also be knowing that this oral polio vaccine is also called as Sabin vaccine. So this is a very good example of this live attenuated vaccine, this Sabin vaccine. It uses attenuated virus. Very important example. This is often asked in the examinations. Okay. Now, what happens in case of oral polio vaccine? This oral polio vaccine, or the virus that you are giving, or the attenuated virus that you are giving in the form of this vaccine, colonizes intestines and induces the production of secretary IgA, IgM and IgG. What will happen? It will cause the production of IgA, IgM and IgG. It will colonize the intestine. Okay, it will colonize the intestine and it will allow the production of secretory IgA, IgM and IgG, and it also requires the boosters. Now you would ask me that you were just saying that only a single shot of this live attenuated vaccines are required. Now you are saying that oral polio vaccine requires multiple shots. Why? Because you see that this oral polio vaccine in basically in India there are three variants of this virus that causes polio. So this shot is basically given thrice in India. This oral polio vaccine is given thrice in India because of the reason being that there are three variants that cause this polio in India. So in each time, it is said that every time the oral polio vaccine is given, it is causing the immune cells to be generated against one single strain at a time. This is the concept. That is why you have to give a booster dose. You have to give the oral polio vaccine 3 times. Otherwise, in all the other diseases, it is just required only one time, or you have to get jabbed only one time. Okay, so this is a very important feature that you should be aware about. Let us move on to the next slide. What are the disadvantages that are associated with this kind of a vaccine? That means live attenuated vaccine. So understand that mutation and reversion is a definitely problem. Now you can see that you have put in a live virus or a live pathogen inside an individual. Obviously it is attenuated, but it can revert. This is what we are saying. Mutation can happen, reversion can happen. Apart from that, it has been seen in case of polio. After administration of the polio vaccine, paralytic disease has occurred, but this ratio is very low. 1 in 2.4 million people. I mean, this is rare, very rare. Only one individual in 2.4 million cases, or 2.4 million people can have this kind of a problem. Now complications, the other disadvantage associated with this the attenuated vaccines is that complications similar to the original disease might occur. Because now you see, measles, when you are giving or administering measles vaccine, the measles has basically measles. When measles occurs originally inside an individual, then also there are encephalitis like conditions. So when you are giving measles vaccine, then also post vaccination encephalitis like conditions are seen. So what I mean to say is that conditions or symptoms that are associated with the original disease can occur once you get vaccinated, or when one individual gets vaccinated. So this is all about the factors, the advantages, the disadvantages, the common principle of the live attenuated vaccine. I hope you would have a clear idea now about this particular vaccine, and in future we would be putting up videos regarding other vaccines also. So stay in touch, and if you like the video, just hit the like button, subscribe to my channel and share it with your friends as well. Thank you so much. Have a good day.